Good morning, good afternoon, good evening, from wherever you're watching. Today's lecture is the biochemistry of nucleic acids. You're welcome to my channel, Dr. HF's lectures. We'll be looking at we'll be introducing biochemistry of nucleic acids. We'll look at what nucleic acids are, what they're made up of. We'll look at the phosphodiester bonds that join them together. We'll study the structure of DNA. We'll look at the forces that denature and stabilize the DNA structure. We'll also take a look at the differences between the DNA and RNA structure. We'll have a look at RNA examples, chromatin, and then we'll end the lecture after that. Nucleotides play very important roles in cellular metabolism, and they make up nucleic acids, and they're chiefly involved in the storage and transmission of genetic information from parent to offspring, which is fundamental for maintaining life. There are two major types of nucleic acids: deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. In 1869, DNA was discovered by Johann Friedrich while he was preparing white blood cells obtained from pores in surgical bandages, and he called it nuclein. If you take a look at the history of DNA, we've come a long way since Friedrich Miescher's experiments. And we can see from 1912 to 1914, William were able to pioneer X-ray crystallography, which laid the groundwork for the structural studies of nucleic acids. By 1920, 1937, we have Phoebus and Florence who were able to capture the first X-ray images of DNA, and William Astbury also attempted an early structural model of DNA. It wasn't until 1944, 1952, and 1953 when we had the likes of Avery, Rosalind Franklin, Watson, and Crick, who were able to prove that DNA carried genetic information and then provide an X-ray diffraction image of DNA, and then publish the double helical structure of DNA respectively. By 1996, Dolly the sheep was the first mammal cloned from a non-embryonic cell, and in 2003, the Human Genome Project was completed after 13 years, where the human genome was mapped, the entire human genome was mapped. So this shows us that we've really come a long way, and we've made giant strides in the discovery and the structural elucidation of DNA and RNA and nucleic acids in general. So just to define some of the terms before we go in, a gene is a stretch of DNA that carries instructions from the DNA. So basically, it carries instructions to produce a functional protein or a functional RNA molecule. And because there are thousands of genes, this ensures, or this means that the DNA molecules can be very large. And the sole responsibility of DNA is to act as a store of biological information and also to transmit this information to produce functional proteins or functional products, as the case may be. RNA molecules serve a wider variety of roles, and there are three major types of RNA. There are other types, but these are the three major types. Ribosomal RNA, which is involved in protein synthesis. Messenger RNA that carries the message or the genetic code from the DNA to the ribosome. And also transfer RNA that brings the activated amino acids to the sites of protein production. So this is just an overview, a schematic overview of the structures of DNA and also the structure of RNA. So basically, DNA is it serves as an archive to store genetic information, while the RNA serves other purposes. From this structure, you can see that there's an mRNA, there's the tRNA, and also there's the ribosomal RNA, which are all very involved with protein assembly in various forms. Nucleotides serve various functions, which can be divided into three major parts. They help to store information and transfer the information. They serve as the universal energy currency, and also in terms of dynamic signaling and catalysis. So nucleotides are the monomeric units of RNA and DNA, and they are the repositories of genetic information and ensure that the information is transferred properly and ensuring that there is high fidelity transmission across generations to prevent loss of information. They're also used as primary energy sources, particularly ATP, which is the universal currency of energy in the body. And they also operate as second messengers in terms of cyclic AMP or cyclic GMP. They serve as coenzymes, which include NAD+, FAD, and coenzyme A. So they serve a diverse range of functions in the body. Nucleic acids are basically made up of DNA and RNA, and they have very strong chemical properties, but they also differ in certain areas. RNA molecules vary greatly in size. They range from fewer than 100 to several thousand nucleotides, while DNA molecules, however, are much larger, and they can extend up to several 100 million nucleotides depending on the organism where they are found. And when they are combined with proteins, these long DNA strands form chromosome to ensure that they are packed properly in the cells. And when these chromosomes are stained with dyes, one can even see it under a light microscope. So let's start looking at the structure of nucleotides. The backbone of the nucleotide is built upon the pentose sugar. And this is one of the main differentiating factors of DNA and RNA. For RNA, the sugar has a hydroxyl group at carbon 2, while for DNA, the sugar lacks an oxygen at the same carbon 2. So we can say deoxy, that's how we got his name, deoxy, without oxygen, lacking oxygen. And the presence or absence of this oxygen at carbon 2 of the ribose is very important, because in the absence of this oxygen, it limits the flexibility of DNA. And this drastically increases the chemical stability of the DNA, which makes DNA to be more suited for long-term storage, because it prevents it from from having different types of reactions. Apart from the sugar, the next thing we have are the bases, and these bases are made up of two types: pyrimidines, which consist of a single six-membered ring made up of 4 carbon atoms and 2 hydrogen atoms, and you can see how it is numbered. It is numbered clockwise. It is numbered clockwise. While the purine, and also the pyrimidine, has a longer name, or it is the smaller molecule. The purine, on the other hand, has a shorter name, or it is the larger molecule, and it is made up of a pyrimidine ring and also an imidazole ring. And the numbering, it is numbered anti-clockwise. Both of them are unsaturated. They contain double bonds, and they are both weak bases, and they are relatively insoluble in water at physiological pH. All of the bases participate in hydrogen bonding, plays a very key role in the formation of the three-dimensional structure of nucleic acids. Both classes of bases are unsaturated and contain conjugated double bonds, which give the ring a flat structure and enable them to absorb UV light. The key pyrimidines are uracil, thymine, and cytosine, while the key purines are adenine and guanine. But if you notice, we have 3 pyrimidines, and that tells us that because one is present in DNA and one is only present in RNA. So basically, thymine is essentially a methylated derivative of uracil, and that is why it is often referred to as 5-methyl uracil. These are the structures of the different bases. We have adenine, there's guanine, cytosine, thymine, and uracil. There are also biological freestanding bases such as hypoxanthine, uric acid, and there's also derivatives from plants, from caffeine, theophylline, and also theobromine from cocoa. So these are all the different structures of the purines and the pyrimidines. So this is what a nucleotide looks like. And we have the base, which is the sugar, the ribose sugar. And this is how it's numbered. This is carbon 1, 2, 3, 4, and 5. This is where we have the difference between the ribose and deoxyribose. So for DNA, there's only a hydrogen group, while for RNA, there's a hydroxyl group at carbon 2. Then the base is usually attached at carbon 1. So this is where the nitrogenous base is attached to. So it can either be a pyrimidine or a purine, and in this case, this is a purine. The shorter name, the larger molecule. Then eventually we have a nucleotide has a phosphate group attached on the carbon 5, and this helps to form the structural backbone of the polymer. But when we only have a base and the sugar, it is called a nucleoside. So a nucleoside is a base attached to sugar, and in RNA we have 4, and also DNA we also have 4. We'll look at the namings very soon. And in both cases, the nitrogen atom at position now a purine or position 1 of a pyrimidine binds the carbon at position 1 of the sugar. While a nucleotide has 3 parts: a phosphate group, the pentyl sugar, which is a 5-carbon sugar, and also the base. In RNA, the sugar is the ribose, while in DNA it's the deoxyribose, and they are different bases involved. So with DNA, you only have A, G, C, T, while in RNA you have A, G, C, and U. It stands for adenine, guanine, cytosine, thymine, and uracil. So just to make it easier, sometimes we use only the single letters to depict the bases that are present. These are analog of the nucleotides, and they have also found relevance in medicine to treat various diseases, and examples include allopurinol, fluorouracil, azathioprine, and also other. So we can also check for others as well. So this is how the nucleoside and the nucleotide are named. Remember we said nucleoside is the sugar and the base. So adenine and the ribose sugar is called adenosine. The sugar with the guanine base is called guanosine. And then we have cytidine with cytosine, and then uridine with uracil. For the deoxyribose sugar, we have a deoxy added to the beginning of the name. So deoxyadenosine, deoxyguanosine, deoxycytidine, and deoxythymidine. The same follows for the ribonucleotide, which is the sugar, the base, and the phosphate group. And take note, in this case, we only have one phosphate group, so that's why we're calling it adenosine 5 monophosphate. And because it's bound at the carbon 5, that's why the 5' is here. Or we can also refer it to as adenylate. But when there's 2 phosphate groups, we call it diphosphate. 3 phosphate groups, triphosphate. So that would be ATP, for example, as an abbreviation for this. The abbreviation is AMP. When there's 2 phosphate groups, abbreviation will be ADP, adenosine diphosphate, and so on. So for guanosine 5' monophosphate, abbreviation is GMP. We also have GDP and GTP. Then cytidine monophosphate, CMP, CTP, CDP, and that goes on. For deoxyribonucleotide, we have deoxy added to the beginning of the name, and that's what makes it different. So nucleotides end in osine, while pyrinucleotides end in osine or osine, while pyrimidine nucleosides end in idine. So that's a way that one can easily differentiate them, while the DNA precursors just add the deoxy prefix at the beginning. To form a polymer of a nucleotide or a polynucleotide, we have to form something called phosphodiester bonds are formed. And it is these phosphodiester bonds that link the nucleotides together to form a polynucleotide via a condensation reaction between the 5' phosphate of one nucleotide and the 3 hydroxyl group of the next. So let's take a look. So we have one nucleotide here and the second nucleotide here, and we put them together, the phosphate group, so the 5' phosphate, and the 3 hydroxyl group of this nucleotide, of the preceding nucleotide, react with the removal of water to form a 3 to 5' phosphodiester bond. So what we see is 2 nucleotides joined together with a phosphodiester bond. And because of this, DNA has a direction. So we start from the free 5' end, because here is free, and then there's a free hydroxyl group on the end at the 3' carbon. That is why this is called the free 3' hydroxy end. This is called the free 5' phosphate end. So it's directional. It goes from the 5' end to the 3' end. And that is why when we read the sequence of DNA, we read it from the 5' to the 3' end, and we write it from the left to the right, and we'll also read it from the left to the right. So this sugar phosphate backbone helps to form the structure of the DNA. And what we can see, these are 4 nucleotides joined together, bound with the phosphodiester bonds. So this is the secondary structure. And if you can see, these strands are anti-parallel or anti-parallel. We have one going in the 5' to the 3' direction, and what is a mistake? So one going from the 3' to the 5' direction. So it's anti-parallel. And what we have here, this is the sugar and these are the phosphates. This is forming the backbone. This is the sugar, the phosphate. This forms the backbone of the polynucleotide. And on the inside, there's the bases that are bound to each other. This is adenine, thymine binding to each other. Adenine, thymine, cytosine, cytosine, guanine. And what is holding them together is the hydrogen bonds in between the bases. DNA is double-stranded. So DNA is a linear polymer of 2 deoxyribonucleotide residues linked by phosphodiester bonds. And this was really deduced by Erwin Chegaff's Watson and Crick. So Erwin Chagall took a look at a wide variety of DNA from different eukaryotes and prokaryotes, and he observed that there are equal amounts of purine to pyrimidine ratios in the DNA of all the species, while Watson Crick supported this by showing that DNA is double-stranded and there was a specific base pairing found between the A and the T, and also the G and the C, and this came to be found to be the B conformation of DNA. So just to look at the structure, we'll describe the structure before we look at it. DNA is made up of two polynucleotide strands, with one strand form hydrogen bond with the base of the opposite strand. So guanine pairs with cytosine, adenine pairs with thymine. There are 3 hydrogen bonds in between the GC pair and 2 hydrogen bonds in between the AT pair. And this really accounts for the Chagall's discovery, where he found that the ratio of A to T is equimolar to the ratio of G to C. DNA runs in opposite directions, so we call them antiparallel, and this was really elucidated by the Watson and Crick model. On one end we have the 5' end, on the other end we have the 3' end. And the spacing between the sugar phosphate backbones is uniform across all base pairs, giving DNA a consistent structure. So DNA basically looks like a ladder, but a twisted ladder, with the base pairs forming the rungs and the phosphate and the sugar backbone serving as a support. Each strand is complementary. So if one knows the 5' to 3 strand structure or sequence, then you can easily deduce the sequence of the opposite strand. The hydrophilic backbones remain exposed to water, while the hydrophobic bases are shielded inside, which makes hydrogen bonding stronger. And then there are 2 grooves where chemical groups of bases are accessible. These grooves allow the proteins to bind and to read DNA sequences without disrupting the double helix. So the main conformation of DNA is the bDNA, and it is a right-handed helix and with a diameter of approximately 2 nanometer. The rise of the helix is the distance between the bases is about 0.34 nanometer, while the pitch, which is the distance of a complete turn, is around 3.4 nanometer with 10 base pairs per turn of the helix. The length of the double-stranded DNA molecules is expressed as base pairs, but when we start to have thousands, we can call them what they are called kilobase pairs. E. coli chromosome is around 4,600 kilobase pairs, so this is equivalent to around 4.6 million base pairs, while the human genome contains around 3.2 billion base pairs. Structure of the DNA, the width is around 2 nanometer, the rise is 0.34, so this is the distance between a base pair, while the pitch is mix of around 10 nucleotides, 10 base pairs per helical turn, which is around 3.4 nanometer. So there's a major groove and a minor groove that allows interaction with other proteins. Then DNA is also antiparallel, so it is in opposite directions, 5' to 3' and 3' to 5'. In addition, there's a base pairing which allows it to be complementary. So DNA is also complementary. The base pairs are complementary. In terms of complementation, the DNA base pairs are complementary, where A binds to T, connected by 2 hydrogen bonds, and G binds to C, connected by 3 hydrogen bonds. And this basically obeys the Chagall's rule, where you see that if there were 2 purines, it would be too wide to fit into the 20 Armstrong or 2 nanometer width. If there are 2 pyrimidines, it would be too narrow. But the perfect combination is having 1 purine and 1 pyrimidine, and then this would fit perfectly into the unit, into the diameter of the nucleic acid. So these are the forces that stabilize the double helix. There's a base stacking, which is made up of van der Waals forces. Although they're weak by themselves individually, but when you add it up together, it gives a very strong force, which helps the DNA to be stable. There's hydrogen bonding in between the complementary bases, which provides a significant stabilizing force. Hydrophobic effects where the purine and pyrimidine rings are buried inside the helix away from water, increasing stability. Then charge-charge interactions where the negative phosphate groups repel each other. And this repulsion can be reduced by cations and also basic proteins such as lysine and arginine. The double-stranded DNA is the far more stable configuration. This is usually found under physiological conditions, but there are certain instances where the single-stranded configuration is required, and this will be during replication, transcription, repair, or recombination, which requires that the strand be unwound or separated. And this process is called denaturation. So DNA can be denatured, and this usually occurs in vitro. So DNA can also be denatured by heat or by chemotropic agents such as urea. So as the temperature rises, the base stacking and the hydrogen bonds become weak and then they eventually break and the strands would separate. The melting temperature is the temperature at which half of the DNA is single-stranded, so it loses its double-stranded nature. And this can be monitored by looking at absorbance at 260 nanometer, where single-stranded DNA shows an increase in absorbance. And this plot of the change in DNA versus temperature, absorbance versus temperature, will give you the melting, or will produce the melting curve. So we have the nature being cooperative. The transition to single-stranded DNA from double-stranded takes place over a very narrow temperature range, and this can be shown by a sharp increase in absorbance at the melting point. So it's also cooperative. Denaturation is cooperative. It begins with a short single-stranded bubble, and then really destabilizes and then causes the bubble to expand, and then the helix unzips, resulting in a sigmoidal melting curve. So GC-rich regions melt at higher temperatures than AT-rich regions because they have higher, stronger stacking interactions and stronger bonds. So these stacking interactions are disrupted first, and when they are weakened, the hydrogen bonds become exposed to water and lose much of their force. Denaturation disrupts hydrogen bonds and it separates the strands. So what we are seeing is the melting curve. First of all, this is the DNA at double-stranded DNA, and eventually it unwinds into single-stranded DNA. And what we see here is that DNA that has 35% GC content will melt at a lower temperature than one that has 50% GC content because the temperatures are 65 degrees Celsius and 70 degrees Celsius respectively. And the main reason is because GC pairs share 3 hydrogen bonds compared to just 2 hydrogen bonds in the AT pairs, which means that higher thermal requirements or higher thermal energy is required to break the GC pairs, which requires higher temperatures to denature. Double-stranded DNA conformations have different conformations. There's the B, the A, and the Z, and they exist in different conditions. The most common is the B DNA, while A DNA is found when DNA is dehydrated, and Z DNA is when certain sequences are present. So these are the differences between the various forms of DNA. The B form is right-handed, and it has 10 residues per turn, and this is the classic Watson-Crick model. The A form occurs in dehydrated environments or RNA-DNA hybrids. It's a right-handed helix and the residues are 11 per turn. While for the Z form is left-handed and also there are 12 residues per turn. RNA plays major roles in gene expression and exists in different copies and forms within the cell. There are different types of RNA, but these are the major types that we find. The ribosomal RNA is chiefly involved in protein synthesis and is the most abundant, making about 80% of the total cellular RNA. The transfer RNA are short molecules that deliver activated amino acids to ribosomes or to the ribosomal RNA during protein assembly, and they are around 15% of the total cellular RNA. And the messenger RNA are the ones that carry the genetic code from the DNA, the translation machinery to specify what protein sequences are required to form functional proteins or functional products. And they make up around 3%. So basically, RNA is single-stranded, but sometimes they tend to form complex secondary structures by base pairing with itself. So one will find where there are short complementary regions forming something called stem loops, where double-stranded stems are produced and they resemble the A form of double-stranded DNA. And they're actually very, very common and widespread due to the various functions that they perform. This is the structure of the ribosomal RNA made up of the 60S and the 40S subunit. The transfer RNA that carries activated amino acids, so this is where it attaches the amino acid and it helps to translate mRNA into amino acids. And then finally we have messenger RNA that is made up of the 5' cap and the poly A tail. The cap is 7-methylguanosine triphosphate that helps to prevent hydrolysis of the messenger RNA and ensures that it remains stable during transportation. Don't worry too much about this. This would be covered eventually when we're doing translation. So this is the acceptor arm. This is where the amino acid binds, and this is the anticodon arm, where there are 3 specific nucleotide bases that recognize and pair with the mRNA codon. So like I said, don't worry too much about this. I was just trying to show us the stem loop structure that RNA forms sometimes to form double-stranded RNA regions. DNA and RNA are very similar, but then differences exist in the structure. So for the pentose sugar, we have ribose and deoxyribose present in DNA and RNA. There's thymine in DNA while uracil is found in RNA. DNA is double-stranded while RNA is usually single-stranded, but with certain times we find it folding against itself. The Chagas rule is strictly obeyed in DNA, while it's not always obeyed in RNA. It is resistant to alkali susceptibility because it lacks the hydroxyl group, while this is susceptible because of the presence of the hydroxyl group. While Urshinol color reaction can be used to identify RNA, and this does not happen with DNA. In 1879, Walter Fleming noticed banded material in stained eukaryotic cell nuclei and called it chromatin, which meant color in Greek. So chromatin refers to DNA that is packaged with proteins in eukaryotic nuclei, and this helps you to compact the DNA. There are thousands of genes which will result in thousands of base pairs of nucleotides, and this has to be packed into the cell, and that is where chromatin comes in. Prokaryotic DNA doesn't have these proteins complexes, these protein DNA complexes, so that's where it differs and are not generally referred to as chromatin. The principal protein in chromatin are called histones, and there are 5 types: H1, H2A, H2B, H3, and H4. And the proteins that are present are basic proteins rich in lysine and arginine, which provide the positive charges. that enable it to bind tightly to the DNAs that are negatively charged, so the sugar phosphate backbone that is negatively charged. And except for histone 1 or H1, all the other histones are usually conserved across eukaryotic species. So for example, the bovine H4 differs from the PH4 by only 2 amino acids out of 102, which indicates that they are the histones, the other 4, are highly conserved. When chromatin is placed in a solution with very low salt concentration, it relaxes and extends. And when it does that, under the electron microscope, it looks like beads on a string. So the beads are referred to as nucleosomes, which are DNA wrapped around histone proteins. And the string is the double-stranded DNA that links them together. So the fundamental repeating unit of the chromatin is made up of nucleosome containing one H1 histone and 2 molecules each of H2A, H2B, H3, and H4, and around roughly 200 base pairs of DNA. The histone octamer is made up of H2A, H2B, H3, and H4, and the DNA that wraps around it. Histone H1 is often called the linker histone, and it binds the linker DNA and the nucleosome core, and is often absent in the relaxed beads on a string conformation, but it is very crucial for driving the formation of higher order chromatin structure to ensure that the DNA is further compacted for efficient storage in the nucleus. So this linker DNA refers to the DNA found between the particles and is usually around 54 base pairs in length, while the nucleosome core particle is the histone octamer and is in close contact with around 146 base pair of DNA. So the octamer is these histones and the DNA, which is around 146 base pairs, while the linker DNA is around 54. So altogether, we have around 200 base pairs of DNA. This nucleosome organization is very important because it not only compacts the DNA, it also ensures accessibility, or it regulates accessibility, because it controls how DNA is wrapped. It influences transcription, translation, replication, and also repair. And the positioning of the H1 and the linker DNA play key roles in determining whether a DNA gene is accessible or not. So this is what it looks like. This is DNA helix that is packed around the nucleosomes. So these are the nucleosomes H2A, H2B, H3, and H4, which then forms chromatin and eventually chromosome. Bacterial cells we mentioned lack histones, so they pack circular chromosomes into nucleoids using polyamines. Histone acetylation and deacetylation are chemical modifications of histone proteins that play a very vital role in regulating how tightly DNA is packed in chromatin. And it also tells how accessible the genes are for transcription or translation. There are 2 processes involved: histone acetylation, and the enzyme is histone acetyltransferases. And what it does is to add acetyl groups to the lysine residues, which then neutralizes the positive charges. And because the positive charges are neutralized, the chromatin loosens because the bond between the positive and the negative charge of the phosphate and the lysine has been relaxed. So the chromatin loosens and the genes become accessible, activating gene expression. While for histone deacetylation, the enzymes involved are histone deacetylases, and they remove the acetyl groups, restoring the positive charges on the lysine residues, which allows the chromatin to condense, and then the genes become inaccessible. So what we looked at the biochemical structure of nucleic acids. We've looked at the differences between nucleosides, nucleotides, the structures. We looked at the structure of DNA, we looked at RNA and the differences between them. We also looked at chromatin. And that's where we're going to stop this lecture. So if there's anything else you want me to add, please let me know in the comment section. Don't forget to like and subscribe for more content like this. So I'll see you in the next video. Bye.